Hi, I'm Laura Mariani from the University of Michigan, and I'm happy today to present to you on multiscalar information, developing a data analysis framework for the molecular analysis of kidney diseases. So I'll start by sharing my disclosures, which are listed here. And the way I'd like to frame this talk is first to start out with a discussion of the challenge of the clinical heterogeneity of kidney disease. And then we'll talk about how molecular analysis of kidney tissue in particular can maybe help to point us to a refined diagnosis, a more accurate prognosis, and effective therapies. And lastly, I'll end with some tools, data, and resources. So let's start off here talking about some clinical information. So I think, across many kidney diseases, we've known for a long time that the current way we assign a diagnosis is really insufficient to accurately predict prognosis and response to therapy. So for example, two of my own patients, they're both women in their mid-50s who've had diabetes for over a decade. Neither one of them has symptomatic neuropathy or retinopathy. Their A1Cs have not been optimally controlled, yet patient A is spilling a lot of protein in her urine and her kidney function is steadily declining. Whereas my other patient has always had relatively low amounts of protein in their urine and has had a stable creatinine for many years. And I think even in promising clinical trial data, for example, this SGL2 inhibitor trial in diabetic nephropathy, in patients who received the active therapy, you can see that for some patients, even in the treatment arm, there are still patients who are reaching the renal endpoint. And I think highlighting the heterogeneity that we observe in the clinic. And so part of what's so challenging, I think, in the clinic is that we have a really limited, vocabulary to describe our CKD diagnoses. And so in the Kidney Precision Medicine Project, one of my favorite parts is that each patient undergoes an adjudication. And so in the CKD Adjudication Committee, which is led by Dr. Poggio McMahon, it brings together a group of clinical investigators and pathologists. And the goal is to really assign a CKD diagnosis. So the group reviews all the clinical and laboratory data up to the time of the biopsy. We then, as a group, review not only the kidney biopsy, but any additional clinical information that's happened since that time. And so on the first adjudication, what often happens is that we do use our standard categories, such as diabetic nephropathy, hypertensive nephropathy. The investigators and people that participate here also rate their certainty, and often there's a low level of certainty on what we think we'll find on the biopsy. And what's so fascinating is that after reviewing the pathology, I think it really highlights that although we do use some standard definitions like diabetic nephropathy and hypertensive nephropathy, there's almost always an observation that there's much more that's observed in the tissue that either has clear or unclear significance. So there have been cases where undiagnosed glomerular disease was found, but also a wide range of other pathologic features that is really unclear what it means in terms of diagnosis or prognosis. And so I think this is really the opportunity of the landscape that we're living in now, that we have all of this high dimensional data that can be generated from human kidney tissue. And that, along with non-invasive biosamples, can really allow us to potentially redefine our classification system to better align with underlying biology. That these tools can allow us to really comprehensively characterize the tissue and provide more granular prognostic information to our patients. And that using some of these novel technologies, we may be able to identify therapeutic targets that are relevant within specific patient populations or across kidney diseases. And so I'd like to give you a couple examples today of how molecular analysis may help us achieve those really lofty goals and improve the care we're providing to our patients. So I'll start here with an example from the Kidney Precision Medicine Project. So we had a patient that was enrolled under the hypertension cohort. So this patient did undergo a research biopsy. It was not a biopsy for usual clinical care. And like many patients I take care of, and I'm sure you take care of, he was a middle-aged man who'd had over 10 years of hypertension. He did not have protein in his urine and had slowly progressive CKD, currently stage 3A. And what we saw first on his biopsy, I think to the surprise of the adjudication committee, despite not having any signs of acute kidney injury, his tubules actually showed some evidence of injury. In addition to that, there were a variety of other features that were found. So for example, on electron microscopy, there was patchy loss of brush border. There was also some ischemic wrinkling in the glomeruli. There's actually a tip lesion that was noted of unclear significance in this patient without proteinuria. And so then we have the advantage of all of those tissue interrogation sites having access to this research biopsy to apply their novel technologies. And so, for example, the group at IU and OSU used their cytometry techniques to use their markers to define not only cell types, but cell neighborhoods, where you can see which cells are next to each other. And for this particular biopsy, the group noted that there was significant immune cell infiltration throughout this biopsy. The same group does laser capture microdissection of different nephron segments as well as compartments in the kidney, and does regional transcriptomics and proteomics. And here again, they could confirm not only that there were markers of tubular injury and immune cell activation, but they could see it in both their transcriptomics and proteomics data sets. The group here that had access to single cell technology generated single cell transcriptomics. And looking at these different cell types, they could also see an increased expression of injury markers in a variety of the nephron segments. And lastly, this group that does single nuclear transcriptomics from UCSD and WashU, they were able to find similar patterns seen across these technologies, and specifically different tubular subsegments that appeared to be injured. And so I think the real power of this approach is that these complementary data sets really offer different windows into the actual biology that's happening within the tissue. And so that can allow you to cross-validate and check that what you're seeing is actually being reflected in the other data types. So for example, this paper describes an approach to data integration using multiple different data sets. So for example, you could imagine starting with single cell data, and that from that you could understand specific cell functions and pathways that are relevant within specific cell types. That then you can then use laser microdissection, proteomics and transcriptomics to assign those cells to a specific region. That you can use other technologies like spatial transcriptomics to confirm that the cells that you've identified in a dissociated fashion are placed appropriately, and that you can use proteomics and metabolomics data sets to see if the processes that are being activated or being repressed are actually reading out in those other data sets. And by combining that with our clinical data and our pathology data, we get to a just much more comprehensive look at that tissue that may be leading us to a better prediction of disease course for an individual patient. And so let me show you some early work that's being done in KPMP following this framework. So here we will start off with single cell data, although you could start with a number of different data domains. So here you can see that this is an early data set. So this data set included 11 patients with acute kidney injury, 10 with diabetic kidney disease, although for one patient, we have 3 technical replicants resulting in additional data sets, and 3 patients with what clinically was called hypertensive chronic kidney disease. So all of these patients underwent single cell transcriptomics of their research kidney biopsy. Those cells were clustered and to identify specific cell types. And you can see the integrated analysis of all 26 data sets here, which identified multiple different cell types and subtypes within those parent groups. So next, what you can do is you can then analyze the list of genes that's being used to segregate those different groups. And you can analyze that list of differentially expressed genes. And you can ask questions like, did we identify pathways that describe the known physiologic cell functions that we assigned those clusters to? And additionally, if there are cells that are here that are falling in subgroups or appear to not follow the expected biology, are we perhaps identifying pathways that represent disease? So for example, here you can see that for the different cell types, we looked at those gene lists and performed an enrichment analysis to see which biological processes are represented by each unique cell cluster. And you can see that the proximal tubule cells have many pathways relevant to energy generation, whereas multiple nephron segments, of course, demonstrate pathways relevant to ion and small molecule resorption. So once we have confidence that those cell cultures those cell clusters are representing unique biological processes, we can then use that information and apply it back to the patient level. So for example, each cell type in that clustering algorithm had between 1 and 9 subclusters within the cell type. And so for example, if we just look at subclusters of proximal tubular cells, we can cluster patients based on the cell type abundance. So for example, you normalize to the total number of cells from an individual patient's biopsy, and you see what proportion of their cells fall within these subtypes. And applying that clustering algorithm, we can see that there were 2 main groups. Cluster 1 contained a subtype of proximal tubular cells that largely was unobserved in patients who fell into cluster 2A and 2B, save for this one patient here. And I will note that those 3 replicates all ended up here, which is good since they're repeating all from the same patient. But you can see as you scan through here that actually these clusters grouped patients very differently than their typical clinical diagnosis. So AKI, diabetic kidney disease, and hypertensive kidney disease were mixed together. And so you can see, for example, cluster 1, like I said, had that unique cell type. Cluster 2 and 2A and 2B were split based on the relative abundance of the remaining proximal tubular cell type. And we can actually use that to look across other cell types. So for example, when we did the same process using subtypes of the thin ascending limb and the thick ascending limb, again, patients who were in that initial cluster based on their proximal tubular cell types, again, they clustered together using these cell types. Note that it wasn't true of every cell type, so when they looked at other cell types, those patients didn't always cluster together, highlighting that biology may be different across the different subsegments. But what then you can do is to ask whether these cell subtypes are associated with particular functions. And so, for example, you can see that cluster 2B had a higher proportion of proximal tubular subtype 3, and that's associated with a variety of transport pathways here. You can see that these different subtypes give us some sense that there is different biology being captured and that different patients do have a different abundance of these different cell types. So there's clearly much more work to be done. This is early data. Certainly the clusters will be refined as there are more samples available. This clustering can be validated using our other data domains. And then whether we can extract and make this relevant to an individual patient as opposed to a group of patients, I think is the ultimate goal. Because in the end, what we would like to be able to do is give our patients information about prognosis and therapy. And so KPMP is obviously very early in its enrollment and does not yet have the longitudinal data that it'll have in the future. But there are many other cohorts that are assembling longitudinal data with the same multiscalar data information. So one of those studies is the Neptune study. So this study is an observational cohort of patients with glomerular disease. And patients are enrolled at the time of a clinically indicated kidney biopsy. The goal was to enroll patients with minimal change disease, FSGS, and membranous nephropathy, and to follow them longitudinally over time. And I think one of the real strengths of the Neptune study is very early on, the pathologists recognized that our current classification system, even within glomerular disease, does not really capture the structural complexity that we observe on these biopsies, and that we may observe a lot more that could be clinically and biologically relevant. And so the pathologists do this really amazing thing with the biopsies. So every Neptune biopsy is converted into a digital whole slide image. And the pathologists go through and annotate each glomerulus at each level. So you can identify glomerulus 1 at each level of the biopsy. And then they go through and score each individual glomerulus for a range of features and mark whether that feature was present or absent at any level of each glomerulus. And so they not only score the glomeruli, but they score the interstitium and the electron microscopy. And what that leaves you with is a really rich characterization of the whole biopsy of these patients. And you can link it to the other data domains that we have in Neptune. So for example, in this study that our group did, we took 221 patients with a usual diagnosis of minimal change and FSGS. And we used essentially the percent of glomeruli that had any given feature. So the percent of the glomeruli for an individual patient that had any one of these features. And we performed hierarchical clustering. And what we found here is that actually, instead of uniquely splitting into just two groups of minimal change and FSGS, we actually found that patients grouped into 3 different clusters. And you can see here enumerating the features that were most predictive of those cluster. And they're actually features that may or may not be typically used to assign a clinical diagnosis. And so when we compared it against our usual classification system, minimal change disease, and then, for example, the Columbia classification for FSGS, you can say that each of our 3 clusters actually contained both patients with minimal change disease and FSGS, telling us that description of the structural complexity is really classifying patients differently. And then, because Neptune does have longitudinal data, we can assess whether these clusters are telling us something about someone's prognosis over time. And indeed, cluster X, that was the one where a lot of the glomeruli had many different descriptors present. Those patients were much less likely to achieve complete remission under usual care, and more likely to lose their kidney function over time. And I think, not only was that true of that cluster, but you can see between clusters Y and Z, they had a different rate of remission and together had similar rates of loss of kidney function over time. So Neptune also has transcriptomic data. So when the patients undergo their clinical biopsy, an extra core is obtained for research purposes, and it undergoes transcriptomic analysis to generate gene expression data. And when we look at differentially expressed genes between these groups, we can see that not only was their pathology different, but their gene expression data was different, and there were unique pathways that were identified for each of these 3 clusters. And so I think really gives us promise that this rich characterization of the tissue, you can uncover potentially clinically and biologically relevant information. So how might we transform these types of data sets into therapies? And is this really just a pipe dream? And I'll argue that I think there's good evidence that this may work and may offer promise for new therapeutic development and better prognosis. So for example, this is a phase 2 trial of baricitinib, which is a JAK2 inhibitor. And there were 129 patients with diabetic kidney disease. They had a mean albumin to creatinine ratio at enrollment of 820. Their mean GFR was 45. And they were randomized to placebo and 4 different doses of baricitinib. And their primary outcome was change in urine albumin to creatinine ratio throughout the study. And so you can see at week 24, the baricitinib groups as compared to placebo had lower proteinuria levels, and that actually persisted after a 4-week washout. So where did the idea for baricitinib and diabetic nephropathy come from? Well, actually it came from multiscalar integration. So for example, this study kind of lays out how that pathway was identified. So comparing human transcriptomic data in diabetics with high and low albuminuria, and then a variety of diabetic mouse models, they were able to identify pathways associated with diabetes and worse clinical phenotype. And by comparing across the mouse models and the human data sets, we're able to identify that the JAK-STAT pathway was potentially relevant. It was followed up with a number of animal model studies, including overexpression of JAK2 in podocytes. Mice were administered, mice with lower STAT3 activity were actually protected from diabetic nephropathy, and multiple other experimental studies to give confidence that intervening on this pathway may be relevant, and resulted in that successful phase 2 trial. So I think what's exciting about this, as you can imagine, this is a hypothesis to be tested. And so the problem with our clinical trials right now is that for many of our kidney diseases, we group what's really a heterogeneous group under one category. And when we go to enroll them in clinical trials, we enroll a mixed population. And so these trials are at higher risk of being unsuccessful. Whereas if we knew that a particular drug was more relevant in a subpopulation, if we could identify those patients and enroll them in a trial that we think has a higher likelihood of success, we may see more successful trials. And certainly this is something to be tested. And so actually the Neptune study in its current phase has a component called Neptune Match. And the idea with Match is that it's testing a process to see whether we can actually communicate this type of information to patients and their providers. And so what happens in Match is that patients are recruited into the observational cohort study. Their biosamples are collected for the usual protocol. They're analyzed to try to identify pathways that are either more active or less active, and as compared to available trials. That information is communicated to the patient, and ultimately when those trials are completed, we can see if patients that receive the stratification information behaved any differently. So I do want to highlight that what Match is particularly studying actually is principally the communication. So can we describe this omics data and these biomarker data results? Can we communicate that to patients in a meaningful way? And then whereas the enrollment in the observational trial is completely separate, the analysis of their biosamples and collection of that is part of the usual observational study. The clinical trials are completely independent, have their own inclusion and exclusion criteria. But the companies that have agreed to partner with Neptune have agreed to share their final trial data so that we can see across many studies if patients have undergone a stratification, did they have any different response rate. And so for example, what would happen is a patient would enroll, there would be an analysis of their biospecimens, and then we would use a statistical model to predict whether or not we think that particular pathway is more or less active in that individual patient compared to the remaining Neptune cohort. And we would offer them a report that would say for a variety of ongoing clinical trials, does their data suggest they're more of a match or less of a match? They subsequently can decide completely independently if they're actually interested in participating in one or any of the trials. So lastly, I want to just share with you some tools and data that could be used for this type of analysis. So there are many online data exploration tools, including the KPMP Atlas, where data is freely available and analysis tools are embedded within these websites to help people analyze this data and test their hypotheses. In addition, there are now many, many cohorts that are assembling these rich data sets, and I think really offer an incredible opportunity for our community as a whole to really change the paradigm of how we analyze kidney tissue. And so I'll just end by acknowledging a huge number of people who are part of KPMP and Neptune, but specifically the patients, the coordinators, and the project managers who keep all of this running and who are really so selfless with their time and their obviously donations of samples to make this sort of analysis possible. Also thank the NIDDK for my own funding.